Chapter 3, Men's Bodies. In this portion of Chapter 3, we're going to be covering male reproductive anatomy. In Part 2 of Chapter 3, we're going to be covering how the various reproductive organs in males work in a lecture titled Male Reproductive Physiology. So let's start first with an overview. Male anatomy is quite diverse with a wide variety in appearance of primary sex organs. This includes penis size, penis length, girth, curvature, sperm production. All of these traits can vary significantly amongst males. Secondary sex characteristics in males include pubic hair, facial hair, and sperm. These secondary sex characteristics develop after males reach puberty. Certain hormones are produced, and they're going to activate and stimulate sperm production, as well as other features that are associated with masculinizing the male body. Here we have a wide variety of different penises. Here we have an uncircumcised penis. Here we have a flaccid penis. Here we have an erect penis. Notice that penis size, coloration can all vary. So now that we've talked about secondary sex characteristics, we're going to spend the majority of our lecture discussing primary sex organs. Two primary sex organs include the gonads and the penis. The gonads are important because the gonads are where sperm and hormones are produced in males. The penis is important because this is a male organ used for sexual intercourse and excretion. Notice in males, the urinary tract and the reproductive tract fuse into the urethra. So rather than two openings as in females, males have just one opening to excrete sexual fluids and also to excrete urine. Now the penis consists of a number of components. First is the root. The root is a portion of the penis that extends internally into the pelvic cavity. This leads to the shaft of the penis. It's approximately 2 inches, and it consists of 2 diverging crura. This is made of both corpora cavernosa and corpus spongiosum attached to the pubic bone by a ligament. It's important to note that both males and females have corpora cavernosa and corpus spongiosum. In males, these are found in the penis. However, in females, the corpora cavernosa and the corpus spongiosum are found within the clitoris. Next is the shaft. This is the length of the penis between the glans and the body. The shaft will eventually lead to the glans or the head of the penis. The glans is the head of the penis with a distinct ridge called the corona. The glans, the frenulum, and the corona or the coronal ridge collectively make up the penis head. The frenulum is an underside tip of the penis. It's the strip of skin that connects the ventral portion of the glans to the underside of the shaft. The corona is the rim of the gland separating from the shaft. And what's interesting about each of these components, the glans, the frenulum, and the corona, these are the most sensitive portion of the penis, and there's a reason for that. It's because of these tangled knots of nerves. These are referred to as genital end bulbs. And these tangled nerves forming these tangled knots called genital end bulbs, they're in the highest density within the glans, the frenulum, and the corona, making the tip of the penis the most sensitive part of the penis. Here's an overview of many of the structures we've covered. We have the root. Well, the root extends into the body. If the scrotum was pushed aside and a male had an erection, like in this example, the under portion or the perineum would be stiff. It would be engorged with blood and erect as well. That's the root. Next is the shaft. The shaft occupies the length of the penis. The glans is the head of the penis, area of high density of nerve endings called genital end bulbs. Here is the corona, or the coronal ridge. This coronal ridge separates the glans from the shaft. Here is the triangular strip of skin called the frenulum. And then this penis is missing foreskin. However, many males have foreskin over their penis, over the glans of the penis. So the foreskin is a loose fold of skin which covers part of the glans. The foreskin can be retracted back, and the foreskin can be removed through a process known as circumcision. Now, circumcision is where the foreskin is surgically removed. And there are some reasons why individuals may have a circumcision performed. This is more common in Western cultures, such as the United States, and it's even more common among certain ethnic groups. White males are more likely to be circumcised than Hispanic or African American males, and there are often cultural or religious implications to circumcision. One advantage of circumcision, however, is this may increase hygienic benefits. If this fold of skin over the glans is removed, then that can prevent accumulation of dead skin cells, secretions, and even bacteria. So that could minimize infection, and that could also lower the incidence of transmitting diseases sexually. For a circumcision to be performed, a device like this, a perplex device, can be used. Other similar devices include a Plastabel or a Gomco device. They all work in a similar fashion. In order for this perplex device to be used, an individual, they would be sized. Notice this is occurring on an adult male. However, circumcisions are often performed on infants, usually within a day of them being born. So in this instance, a line would be drawn, and the line is drawn based on the World Health Organization guidelines on which area of the foreskin would be removed. And then this device, this perplex device, would be moved to the base of the penis. The foreskin would be pushed up. Next, there would be an inner ring. This inner ring would be placed between the glans and the foreskin. Notice there's a groove on this inner ring, and that part is important because this black portion at the base of the penis is an elastic band, similar to a rubber band. This device will move up, this plastic band will detach, and this will go right along the inner ring, pinching the foreskin. This will pinch the foreskin, and this outer base, or this outer ring, would be removed. And as this pinches the foreskin, within a couple of days, this could cause some of the cells to cut their blood supply off on the foreskin. Some of these cells of the foreskin will die, and then a couple of days later, a doctor could go in and surgically remove any of this foreskin. Other components of the penis include the corpora cavernosa. With the corpora cavernosa, this includes two expandable spongy tissue cylindrical bodies inside the penis. These cavernosa bodies engorge with blood during sexual arousal. These account for the majority of an erectile capacity. That's because they're loaded with sinusoids. Sinusoids are collapsible tissue capable of filling and engorging with blood. Here we have one cavernosa body. Here we have a second cavernosa body on this frontal section of a penis. In addition to the two cavernosa bodies, there's a corpus spongiosum. This is the corpus spongiosum right here. The corpus spongiosum is also expandable spongy cylindrical tissue. However, this surrounds the urethra. This doesn't have a tough, fibrous band of connective tissue around it like the corpora cavernosa do. And it's important that it doesn't, because this spongy tissue, it needs to be able to secrete fluid in the event of an ejaculation. Notice each of these cavernosa bodies are not exactly the same in their size or symmetry, and this could contribute to curvature of the penis. As the penis becomes erect, these collapsible spaces fill with blood. That's going to cause the penis to increase in girth and length, and because arteries deliver blood to the penis and veins are constricted, they don't allow much blood to escape from the penis. The penis fills with blood, filling up these sinusoids, becoming erect. So here you see the vast majority of sinusoids inside the cavernosa bodies, because again, the cavernosa bodies account for the majority of a male's erectile capacity. So now let's take a look at some other characteristics of a male penis. The average length is about 3 to 4 inches in a flaccid penis. On average, males have a penis from about 5 to 7 inches. The average diameter is about 1 1/4 inch flaccid, however, about 1 1/2 to 1 3/4 inch when erect. What determines a man's penis size? Genes. Genes determine the length, girth, and curvature of cavernosa tissue. This cavernosa tissue can affect, again, the length of the penis girth and curvature. This is influenced by genetics. So this is a study conducted by the Kinsey Institute, indicating that 77% of males, in this particular study, the demographic was conducted with white males, but this study indicates that 77% of males have a penis size between 5 and 7 inches. Are there outliers? Yes, there are. However, the vast majority of males have a penis within this range. And this shows the same data, however, in a histogram. The vast majority between 5 and 7 inches. So now let's take a look at these cavernosa bodies and corpus spongiosum from a different diagram, a different perspective. Here we have the bladder, the prostate gland, the bulbourethral gland, and right here is the bulb and the crura. Remember, the bulb and the crura make up the root. The root is the internal portion of the penis. The crura extend into the cavernosa bodies. And right here we have the corpus spongiosum surrounding the urethra. This explains why the underside of the penis is spongy and the glands are spongy even during an erection. Here is an alternative view showing the cavernosa bodies, corpora cavernosa. Here is another corpora cavernosa, corpus spongiosum with the urethra in the center. Although this looks kind of like a happy face, 2 eyes and a mouth, this is not. This little circle right here represents blood vessels. Here we have more blood vessels and more nerves. Here you clearly see the bulb and the crura connected to the pelvis. Our next structure is the scrotum. The scrotum is important because this is a layer of skin and muscle that's going to cover the testes. The scrotum has 2 principal layers, the skin and a smooth muscle called tunica dartos. Tunica dartos is composed of smooth muscle and connective tissue. It's important to note that the tunica dartos in particular is a layer of the scrotum. It's a layer in the scrotum that contracts and relaxes in response to temperature change. It is an involuntary muscle. Males do not choose consciously to contract or relax this muscle. There is another muscle, however, that could influence elevation of the testes. This is the cremaster muscle. And there are two cremaster muscles, and each of these muscles is going to extend down to each of the testes. Although the cremaster muscles are not part of the scrotum, they also can contract and relax, elevating the testes in response to temperature change. So both of these are responsible for the position of a man's testes in response to temperature change. However, the tunica dartos is a layer of the scrotum. The cremaster muscles are not. Why might males have muscles that influence the elevation or the position of their testes in response to temperature? Well, this is very important. The testes are kept about 7 to 12 degrees Fahrenheit below body temperature. If a man's body temperature on average is 98.6 degrees, the testes are kept right around 90 degrees or just below that. There's an important reason for this. If it's too cold, well, that's going to affect sperm production. Sperm production would be drastically slowed if the testes were too cold. So in the event that the testes are ultimately that the male is cold, the testes could be brought closer to the body. They could contract, or I should say the scrotum, and the cremaster muscles can bring the testes closer to the body, so the testes could get heat from the body so they're not too cold, so they could maintain optimal sperm production. If it's too hot, well, that could be problematic, because if it's too hot, this can cause sperm to mutate. That would be problematic because sperm are used to produce offspring. So in the event that a male is hot, sweat glands at the scrotum and in the genital area will release sweat. That'll help cool off the testes and the scrotum, and this can help keep the testes away from the core of the body. If the testes are kept away from the core of the body, they could cool down so they're not too hot, and sperm are less likely to mutate during this process. If sperm are heated up, they could also decrease their lifespan. So this shows the cremaster muscle. We see that this is going to wrap around the spermatic cord, which we'll talk about in a moment. And this can contract, bringing this entire testy up. Here we have this middle layer. This muscle layer right here would be the tunica dartos. And here we have skin. The reason why the scrotum appears wrinkly is because this is excess skin with an underlying muscle. If it's cold, this contracts and this wrinkles up. If it's warm, these muscles relax and the skin would be more flat. So the cremaster muscle elevates the testes, and the tunica dartos can contract, bringing the testes closer to the body. Now the spermatic cord is what the cremaster muscle wraps around. The spermatic cord can be felt. This includes the vas deferens and blood vessels connecting to the testes. This is important to deliver blood and to deliver nutrients to the testes so sperm can be produced and hormones can be transported out. So here we have one spermatic cord on this side, and there would be another spermatic cord on the other side delivering nutrients to the other testicle. Next, we have the vas deferens. Now there are two vas deferens. These are long, thin ducts inside the spermatic cord. And the vas deferens is important because this is the tube or duct that sperm travel through. The vas deferens will start at the epididymis, and they'll end at the base of the bladder where they join with the seminal vesicles forming the ejaculatory duct. Sperm travel to the seminal vesicles by cilia. It's important to note when sperm are produced initially, they're not yet active. Initially, they're brought up from the testes in the vas deferens by little hair-like cilia that are going to draw them up. And then after that, when they're released through ejaculation, chemicals called a fertilization promoting factor as well as other chemicals will activate sperm. Then they can propel themselves, but not yet, at least not initially. The seminal vesicles, there's 2 seminal vesicles. These are important because they help secrete alkaline fluids that will aid in neutralizing the acidic vagina, and the seminal vesicles also produce fructose. Fructose is the energy that sperm would use in order to propel themselves and find an egg. Most of the volume in semen is from the seminal vesicles. Approximately 70% of the volume in semen is seminal fluid from the seminal vesicles. Next, we have the prostate gland. Males have only one prostate gland. After the vas deferens fuses with the seminal vesicles and merge at the ejaculatory ducts, this empties right into one prostate gland. The prostate is made of smooth muscle and glandular tissue, and both the ejaculatory duct and the urethra pass through the prostate. The prostate is important, and it's only found in males. The prostate is important because this produces about 30% of the volume within semen. This produces a thin, milky, alkaline fluid, resulting in a favorable pH or acid level for sperm. This is also going to help neutralize the acidity inside of the vagina. So this is going to be alkaline rather than acidic. It's going to be an optimal pH. Next is the bulbourethral gland. Males have 2 of these bulbourethral glands. These are sometimes referred to as a Cowper's gland. Now with the bulbourethral gland, this is a gland that's going to produce a slippery fluid. If males are aroused, especially if they're aroused for a prolonged period of time, they could sometimes release a little bit of fluid prior to ejaculation. This is known as pre-ejaculate or sometimes referred to as pre-cum. This is fluid from the bulbourethral gland. Now this has an important function. Because the urinary tract and the reproductive tract both merge at the urethra, the bulbourethral gland releases a slippery fluid to help lubricate the inside of the urethra. Remember, if males are urinating through the urethra multiple times per day, that can cause the urethra to become very acidic. This will help neutralize the acidity, so when sperm pass through the urethra, they are not killed by the acids from the urine inside the urethra. This typically doesn't have sperm in it. So this is an overview of many of the structures that we've gone through so far. Here we clearly see the bladder. It's where urine is stored. Urine would pass through the bladder, through the urethra, out the penis. Here we have the testicle. We'll discuss some components of the testy in more detail in just a moment, but let's go through this pathway because this is important. This may come up on your next exam. I could ask you, diagram discuss or identify the correct pathway of sperm production as it's produced and as it travels through the male body out the urethra. It's interesting to note that the testy is located right here, outside of the core of the body. It's produced here, it exits here. It's produced here, but exits right here. But look, this has a very interesting pathway. Sperm are produced in the testicle. They're stored in the epididymis. They make their way up the vas deferens. They're moved by cilia. Remember, they're not active, they're not propelling themselves. Cilia is moving them up. They make their way around the bladder, into the seminal vesicles, ejaculatory duct where they dump off into the prostate. And then from the prostate, when an ejaculation occurs, they're going to be released out of the penis. Why would the body go through such great length to produce sperm in a location that is very close in proximity to where they're released? It's because sperm by themselves aren't sufficient to fertilize an egg. They need ingredients from the seminal vesicles and the prostate. So they must enter into this epididymis, vas deferens, seminal vesicles, and then the prostate before they exit, because the vast majority of semen is ingredients to support the sperm. The sperm itself occupies a very small portion of semen. Here is the bulbourethral gland, also known as the Cowper's gland. This will lubricate the urethra prior to an ejaculation. This is a pelvic floor muscle in males called the pubococcygeal muscle. This is a pelvic floor muscle that can be flexed. This is an important muscle because this aids in ejaculation. Now let's take a look inside the testes. The testes contain a number of important components, and one of them is the seminiferous tubules. Males have anywhere from 250 to 500 seminiferous tubules, and these seminiferous tubules are highly convoluted. They're coiled. They have lots of twists and turns. They form cone-shaped lobes. And with these seminiferous tubules, there is a lot of surface area. Next are interstitial cells. These interstitial cells produce androgens. Androgens are a class of hormones that include testosterone. These are very important cells because these produce the majority of testosterone in males. The location is pivotal because these interstitial cells are located between the seminiferous tubules. They're located very, very close to the seminiferous tubules. The epididymis are two crescent-shaped structures on top of each testis. The epididymis is important because this is the site of sperm maturation, and this is also a storage chamber. Now, sperm can be stored for several weeks. However, if sperm is abnormal, if it's stored too long, or if it's defective in some way, it can often be identified and removed. This is accomplished by tagging sperm with ubiquitin. Ubiquitin is a chemical tag that is 72 amino acids long, and if sperm are too old or abnormal, they're often targeted with ubiquitin so they could be degraded and their constituents can be recycled throughout the body. Here we have these cone-shaped lobes. This would represent a seminiferous tubule. Here is another seminiferous tubule. Here is another. Remember, there's about 250 to 500 of these per testis. Lots of these little coiled, convoluted tubes. This structure is the epididymis. Here we have the head of the epididymis. Sperm would enter here. They would move to the body of the epididymis where they're continuing the process of maturation. And then the tail, where the sperm would be pretty mature by this point. Now they can move up to the vas deferens. Couple of other points to note. If we were to take a seminiferous tubule like this one and cut it, we would be able to look inside and see all these little dots. If this is a cross-section of just one seminiferous tubule, all of these little dots are cells that are dividing to create developing sperm. The seminiferous tubules are the specific site of sperm production. Also, it's important to note, here we have one seminiferous tubule loaded with immature sperm cells. Here we have another surrounding it. Here's another surrounding it. Here's another surrounding it. However, in between the seminiferous tubules are interstitial cells. These interstitial cells are important because, again, they produce testosterone. We're going to see later how testosterone is going to activate some of these cells to divide to create sperm in the first place. Let's go through an overview of many of the structures that males use to produce sperm. Well, here we have the bladder. That's where urine is stored. Behind the bladder, if you can see this structure, this is the seminal vesicle. Remember, there's going to be another one on this side. Here we have the vas deferens. The vas deferens is lined with cilia. That cilia will draw sperm up after it's produced. Here we have the prostate. The prostate produces thin, milky alkaline fluid. Creates a hospitable pH for sperm, so it's not killed by the acidity in the female reproductive tract. Here we have the Cowper's gland, or the bulbourethral gland. This will release a slippery fluid, clearing the path and neutralizing any acid from urine that may be in the urethra. Here we have the epididymis, the site of sperm maturation and storage. And then, of course, the testes. In our next lecture, we'll see how all of these organs work together. So after sperm's produced, it can be released through ejaculation. We'll talk about the hormones and the important organs that play a role in ejaculation in our lecture on male reproductive physiology.